On the last video, I talked a lot about how we can have a signal that's transferred from cell to cell when they're in direct contact with each other, really close, or where they need to get that signal from really long distances. But what we need to look at next is what happens once the cell actually gets that signal. How does it take that signal and actually do something with it? And one example I like to look at for this is like a plant. A plant is going to receive signal from the light in the direction in which the light is coming from, and its cells are going to need to respond to make it so that the plant is actually going to grow towards that light. And so we need to learn how the cell is able to do this. So we're going to look at these pathways that are happening inside the cell to make this process actually happen. And so we're going to start off first with really looking at how we get that signal to it through the process first of reception. So the first thing is we're going to see that there's three stages of this cell communication. And stage one is reception. That is going to be that we have some kind of signaling molecule, maybe that's a hormone, maybe that's some type of protein, that is going to need to be able to be picked up by a receptor that's on the outside of the cell, and so that signal molecule is going to bind to that particular receptor. And so we call this reception. It's detection of the signal from the outside of the cell. Then what's going to happen next is this process of transduction. And in the process of transduction, it's going to be when we're going to convert that signal into a form of something that that cell can actually understand. The cell can't understand what a hormone means unless it is actually going through the process of transduction, and then the last thing that's going to happen is the actual response. That cell is going to respond in some form to the message that it received. So when we look at this, we're going to see that this process is not always exactly the same where we have reception, transduction, and response. It's going to happen this way, but there's going to have to be a few things that are a little bit different about this based on the modifications that we see. And so I'm going to move this up here, but we're going to have protein modifications that are going to happen. And so when these protein modifications happen, we're going to see different things that are going to happen, including the regulate protein synthesis by turning on and off genes in the nucleus. So that's one thing that could happen as a response. We could see that it's going to regulate activity of the proteins in the cytoplasm. We also could see cascades of intermolecular interactions that are going to relay signals from receptors to the target molecule. So maybe that signal was not actually meant for just that particular location, but maybe there's target molecules that it needs to get that information to. And we also will see this concept of a phosphorylation cascade, which is really going to enhance or amplify the signal that's happening there. So when we look at this, signaling is going to begin with the recognition of a chemical messenger, and we call that a ligand. And so that's going to be by a receptor protein on the target cell. So down here we see that this is our cell here, and on this cell there's going to be a particular receptor that has a particular shape. And so we're going to see that it's going to be a recognition of this particular chemical messenger. In this case, it's a hormone, and we call this a ligand when the hormone is going to bind to that receptor on this particular target cell. And so what's going to happen is that ligand binding domain of the receptor is going to recognize this particular chemical messenger. This messenger in this case is a hormone, but it could be a peptide, it could be a small chemical, it could be a protein. And so they have a specific relationship, and you can see how this triangle fits very well into this receptor here. And this is then going to change the GDP, which is the inactive state, into GTP, which is the active state. And so when it's in the active state, the GTP then can send a message here to this cyclase, and this cyclase here is going to then make it so that the ATP is going to be able to activate the CAMP, which is going to actually activate the protein kinases. And so it is a couple steps to this process, but it's going to take a receptor binding, then GDP going to GTP, GTP then binding to our cyclase here, which then activates ATP, which activates our CAMP, which activates our protein kinases. And that then is going to send the signal to other parts of that cell. We call these things G proteins, and they're coupled receptors, and that's an example of a regulatory protein in eukaryotes. When we look at this, we're going to see that there's going to be signaling cascades that are going to relay messages from the receptors to the target cell. And we kind of looked at this already, But we're going to see this when a cell needs to grow, when it needs to secrete molecules, or even when we see gene expression. And we're going to be going into way more detail on this in the next couple chapters. But just a quick refresher one more time, we see here that there is going to be a ligand that's going to bind to the intracellular domain of a receptor protein, and it's going to change shape. Pretty much all you need to know. Hormone is going to change the shape of our receptor once it binds. What's going to happen then is GDP is going to turn to GTP, which is going to activate this cyclase here, which is going to make it so that ATP is going to convert it into a secondary messenger of CAMP. These are secondary messengers that are going to relay or amplify the signal to the rest of the cell, and it's going to have an effect on the entire cell's function. So, binding of a ligand to ligand gated channels can cause the channel to open or close. And so, these need to bind to that particular channel. Signal transduction pathways are going to look different though if it's a protein or if it's a steroid. With a protein here, we are going to see that this protein is going to bind to the receptor protein here, which is then going to have the secondary messengers in the cytoplasm that are going to be able to move on to the nucleus. It is different for a steroid. A steroid can actually move right through that target cell membrane and it can go right through into the nucleus and end up as a protein in the end. For a protein, though, it's a much harder process that's going to happen because you need a receptor protein and you also need a secondary messenger as well. And so keep that in mind when you're looking at this because that steroid can move through the cell membrane really easily, where the protein is not. It's too large and it's not able to move through, so it needs a receptor there. I am going to give you this example if you would like to take a look at it. And then if not, you can just kind of move the video to the end if you would not like to look at the AP questions. But what it says is metformin is a drug used to treat type 2 diabetes by decreasing glucose production in the liver. AMP is a major cellular regulator of glucose metabolism. Metformin activates AMPK in liver cells but cannot cross the plasma membrane. By blocking AMPK with an inhibitor, researchers found that AMPK activation is required for metformin to produce an inhibitory effect on glucose production by cells. Okay, there's a lot of information there. Let's break it down. Which of the following best describes the component that metformin represents in a signal transduction pathway that regulates glucose production in the liver? Even with all that information here, the really important thing to look at is understanding that the things that we're talking about when we're talking about these protein kinases are looking at ligands and them being able to bind, and then looking at secondary messengers. And so we remember from the last slide that we know that that protein is going to need to bind to the ligand part at the receptor, and we call that a ligand, and that's going to activate then the AMPK to be able to make it so that next process were to happen. So the first answer says it's a secondary messenger that amplifies a signal through a cascade reaction. If you go back here, we actually see with our proteins, I'm going to go back to this one here, that it's going to need to be able to bind to a receptor protein first before it can have a secondary messenger there. And so it's this ligand binding to the receptor protein that's really important there. So the answer should be B for this one. A few key takeaway points that you're going to want or to have in your notes is going to be these. The first one, a signal transduction pathway is the binding of signal molecules to receptors. So we know this. There's going to be some type of thing that is going to be a molecule that's going to bind to a receptor, usually on the cell surface or inside the cell, and it's going to signal a response by that cell. Cells are going to use these pathways to link signal reception with cellular response. Things like growing, secreting things, gene expression are all going to be really important. A pathway is going to begin when a receptor slash ligand binds to an external receptor or an intracellular receptor. We looked at that. Then what we're going to see is that the protein modification is going to cause a conformational change to where the ligand binded. And this change is going to send an intracellular response, which is going to cause a secondary messenger to be activated. And so that's when we saw that GDP being activated to GTP, which then activated our secondary messenger of CAMP. And then what we are going to see at the last one here is a phosphorylation cascade is a signaling pathway where one enzyme is going to phosphorylate another, causing an amplification of the reaction. And again, we saw that when GDP went to GTP, and through this phosphorylation event, we saw that it's going to amplify the reaction. And so thousands of other proteins are also going to be able to go through that process. That is it for understanding signal transduction pathways. We will be doing lots of practice with this in class as well.